Hi everyone, this is Chapter 1, Part 3 of Cell Structure, where we'll be talking about cell structure and function, mostly organelles, really. Now, in cell structure and function, first of all, we kind of need to define a few terms. When we say organelles, what do we mean? We usually mean functionally and structurally distinct part of a cell that's surrounded by membranes, that's like separate from one another and has a separate specific function, like say the nucleus from the rough endoplasmic reticulum. These are surrounded by membranes and they are like separate functions. Now, ultrastructure is kind of more general, is not necessarily surrounded by membranes. Sometimes it's just detailed structures of a cell, kind of like the cell membrane. The cell membrane, we don't usually look at it as an organelle. It's a cell surface membrane around the cell. It's an ultrastructure instead of an organelle. So yeah, now, under cell structure and function, there are actually a few things we need to talk about. There are prokaryotes and eukaryotes. Okay, the whole world, all the organisms in it can be mapped onto a tree of life. And there are two major types. Prokaryotes, which consist of bacteria and archaea. Later on in this chapter, we go into bacteria a little bit. But right now, we are looking at eukaryotes, where we are at. We are animals. We are somewhere in this category of mammals here. And there are plants under eukaryotes as well. And we are talking about these types of cells first. Eukaryotic cells with eukaryotic cell structure first. Now, talking about eukaryotic cell structures, plant and animal cells included, you need to do three main things. Number one, you need to name it when you see it. Okay, in any diagram, you need to now name it. Number two, you need to recognize it from any diagrams or micrographs they may throw at you. And number three, you need to know its functions. There are quite a few eukaryotic cell structures you need to know about. These are the 16 types. And today, we'll be covering 9 types, 1 up to 9. Let's go. Number 1, there is cell surface membrane. Again, cell surface membrane is not really an organelle. It's like an ultrastructure. You can see under the electron microscope, it kind of looks like this. It is also called the plasma membrane. It is around 7 nanometers thick. Again, being able to see it under the electron microscope, not so much the light microscope. And under the electron microscope at 100,000 times, it will be seen as 3 layers, and therefore we say that it gives trilaminar appearance. Now, the appearance is due to this phospholipid bilayer it is made out of. So it looks like a sandwich, perhaps like 1 layer, 2 layer, 3 layer kind of situation. But really, it's a phospholipid bilayer. There's only 2 layers of it. This is one phospholipid. This is a phospholipid bilayer. Anyway, it's partially permeable, so not all substances can freely move in out the cell because its function is to be on the outside and control movements of substances into and out of the cell. You will be learning a lot of detail about the cell surface membrane and its structure in Chapter 4. Moving on to the next organelle. Nucleus. The nucleus is a famous organelle. You should know this by now. But did you know it is the largest organelle and the most visible when it's present under a light microscope? It has double membranes, by the way. This means it has 2 phospholipid bilayers. Two. And well, its general function really is to contain a lot of genetic information and keep it kind of separate from the cell because the cell may have a lot of reactions. So the nucleus kind of contains this DNA, genetic information for what? For the synthesis of protein. Now, the DNA inside the cell would be transcribed into mRNA. That is a new word for you. it undergoes a process of transcriptions. It gets converted to mRNA, and this mRNA is then translated into proteins later on in the cell, not in the nucleus, but somewhere else in the cell. Now, this DNA, as I mentioned just now, is in the nucleus and protected inside. Now, that's not it, okay? That's not it. This is a general function. But within the nucleus, there are also more ultra structures besides the nuclear envelope, which is the membrane that is kind of outside the nucleus on the surface. You must know that this is attached to ER, by the way, ER, endoplasmic reticulum, and is made of two membranes. Again, two phospholipid bilayers. This nuclear envelope also has nuclear pores, by the way, and these pores are how substances move out and in of the nucleus and cytoplasm. So it's kind of like the cell surface membrane, but for the nucleus. Controlling movement of substances is incredibly important because it controls function, right? Controlling movement means controlling function. There are proteins, there are mechanisms in place to direct that control. Anyway, that's the nuclear envelope that has more detail than I thought it would have. The second part is the nucleolus. Nucleolus is the densest region of the nucleus. And really, it's the site of ribosomal RNA synthesis. So other than normal messenger RNA, the nucleus also produces ribosomal RNA, which, you guessed it, is used for ribosomes, right? It is the site of ribosome assembly. Ribosomes are assembled and then it can be used later in the cell. Last but not least, there is chromatin. And chromatin is basically what we call DNA and its proteins. Because I know we imagine DNA as this double helix, but honestly, in the cell, DNA is often associated with proteins. Means, imagine a double helix, they are kind of wrapped around proteins called histones. And you'll be learning more about histones and DNA packaging later on in your A-levels as well. Specifically, it's in chapter 6. Anyways, that is the nucleus for you. There's the nuclear envelope, there are nuclear pores, which is part of the nuclear envelope, nucleolus, and chromatin. Let's talk about ribosomes next. Well, as we know, the nucleus has DNA, it gets transcribed into mRNA, and the mRNA actually exits the nucleus, and it actually goes into the cytoplasm. The cytoplasm, in the cytoplasm, there are ribosomes where translation occurs and converts, right, translates that mRNA into protein. Ribosomes, as we know from previous videos, is the smallest organelle. It is only 25 nanometers in size. It is not bound by a membrane and is one of the only organelles not to be bound by a membrane. This is the exception. Now, it's made of rRNA, which is synthesized in the nucleolus that we saw just now, and also some other protein. It has two subunits, as we can see here, a large subunit and small subunit. It's usually simplified diagram in this manner here. Now, its function, as we can see in this picture and my explanation just now, is to produce protein. It is the site of protein synthesis. And you can see here how it can also sit on the rough endoplasmic reticulum. You can see how the mRNA passes through it, and the amino acids are assembled into a protein, a polypeptide chain, a protein in a certain way. It's pretty cool, right? Now, there are two types of ribosomes actually. The ones that I've been talking about is the ATS ribosomes. They are 25 nanometers. This is the normal ones in the cytoplasm and rough endoplasmic reticulum of all eukaryotes. However, there are such things called 70S ribosomes, and 70S ribosomes are smaller. We don't remember the size here. You just need to know they are smaller, and they are found in the mitochondria and chloroplasts of eukaryotes. Can you believe it? Right, there's ribosomes in your cytoplasm in rough endoplasmic reticulum, and there's also ribosomes, but a smaller one, a different type, in mitochondria and chloroplasts of the eukaryotes. And of course, in prokaryotes, they also have 70S ribosomes. They are just a bit smaller. S is actually a kind of unit for size. Let's leave it as that. Number 4, endoplasmic rough endoplasmic reticulum, more commonly known as RER, because I'm too lazy to say the whole thing. It's a mouthful, okay? As we know from just now, it's an extensive connected system of membranes that's actually connected to the nuclear membrane. Now, its extensive connected system of membranes that is connected again with the nuclear envelope is called cisternae. Cisternae. Cisternae means flattened membrane sacs, and don't you think it looks like that? It's like membrane squish, like long, then they became longer. Anyways, it runs through the cytoplasm, and it has a rough appearance because there is ATS ribosomes attached. So clearly, its function mainly is for protein synthesis, but the rough endoplasmic reticulum also can help in protein modification to some extent. Very simple ones like protein folding or glycosylation, which is the addition of carbohydrate chains to protein, which is actually a very common process to modify your proteins. Yeah. Energy. They also transport this protein after it's synthesized to the Golgi for more modification. Now, let's talk about its sibling, smooth endoplasmic reticulum. What gives it that appearance is the absence of ribosomes. Now, it's continuous also, by the way, with the rough endoplasmic reticulum. And you can see it in this three-dimensional really cool picture here that they are really all connected. The nuclear envelope, the rough endoplasmic reticulum, the smooth endoplasmic reticulum. We just get a cross section really, but really they're all connected. Anyways, what is function for? Now, rough endoplasmic reticulum is mainly for the synthesis of protein, but smooth endoplasmic reticulum is mostly for lipid and steroid synthesis. Examples of this include cholesterol, steroidal hormones, etc. So yeah, that's all you need to know. Now back to the protein bit. So RER, rough endoplasmic reticulum, like synthesizes proteins and it modifies them a little bit sometimes and it transports it to the Golgi, right? So let's talk about the Golgi. My students call this Wi-Fi. It doesn't look like Wi-Fi, a little bit like inverted. But anyways, the Golgi body is a little bit separate actually from the ER. So it's a little bit further from the nucleus. That's how you kind of guess that it's the Golgi body and the electron micrograph. It's also made of cisternae like ER, which is flattened membrane sacs, but again, separate, discontinuous from the nuclear envelope, the endoplasmic reticulum, and so on and so forth. Now, what is special is also there's no connection between members of itself. So unlike endoplasmic reticulum, these members are kind of separate-ish, and it has swellings at the end of the sacs for vesicle formation. This is because it's constantly being formed and broken down by these vesicles. It's being formed by transport vesicles from the rough endoplasmic reticulum on the cis phase, and it's being broken down to form continuously to form secretory vesicles and lysosomes on the trans phase. And what this means is that it's forever like evolving and moving. Now I like to like kind of describe it to my students as like the what microbots in Big Hero 6. I don't know if you watch Big Hero 6, but basically it's like a bunch of tiny robots that can become whatever they want, and they can fuse and they can split from each other, and they can make monsters, or they can make like separate tiny monsters. I don't know. But they're quite useful. And actually, the Golgi characters kind of functions like that. Basically, all membranes function like that. Because if you think about it, all membranes are made of phospholipid bilayer, right? And this phospholipid bilayer can kind of like bud off and form something on its own, but it also willingly fuses and forms and merges to form something else. So this is kind of like that. Transport vesicles like fuse to form part of the Golgi, and part of the Golgi kind of buds off, breaks down to form other vesicles and lysosome into smaller components in order for other things. A little bit more about Golgi body. Let's talk about the function. So why is it like constantly budding off and like fusing in? What is the function? Number one, it is for modification mostly of both proteins and lipid. For example, glycosylation. Okay, some overlap in function with the endoplasmic reticulum. That's okay. Our body loves redundancies, like basically like overlapping function. That is okay. Phosphorylation, addition of phosphate groups, that is a very common modification as well as the cutting and folding of proteins. So the Golgi bodies would probably have codes, enzymes in order to carry these things out. Next, packaging molecules into vesicles for transport. So the proteins come from the ER to transport vesicles, and then after it's modified, maybe these molecules can be packaged. Okay, this protein or lipid can be packaged into secretory vesicles for release of protein maybe out of the cell, or also secretory vesicles to place proteins at the membrane of the cell. Other than that, there's also a budding off to form lysosomes, which contain proteins. Those proteins are enzymes called lysozymes. We'll talk about that later. Now let's look at an overview of what's going on. We can see here how proteins are made in the RER, right? And these here are transport vesicles. Can I write that? Does it like it if I write on the animation? Hey, I can. Okay, so the transport vesicles actually bud off from the ER, carrying those proteins, and you can see it fusing with the Golgi body. And in the Golgi body, it's being modified so it becomes red. Here, again, it's just a diagram. And it's packaged into secretory vesicles to be kind of transported out of the cell. And this is a process of exocytosis. So I hope you can imagine this flow. Let's look at another image of this, right? This is a label image. You can see here again, the proteins form in the rough endoplasmic reticulum. Bear in mind, proteins form, as in polypeptides form. Amino acids are not formed, right? Amino acids are kind of joined together to make proteins. Anyways, proteins are synthesized in the RER, transport vesicle from the ER, goes to Golgi apparatus, goes to secretory vesicles, and that secretory vesicle basically fuses with the plasma membrane in a process called exocytosis. You can see here, and it fuses and the protein gets released out of the cell. This is such a cool animation here because it really shows how the microbots, basically the phospholipid bilayer, is able to fuse, and basically this vesicle can become part of the cell surface membrane. Now I usually get the question, Miss, if there's a lot of exocytosis, will it make the cell bigger? Because the cell surface membrane kind of adds, right? It adds to the cell surface membrane. And the answer is very possibly yes. But don't forget, the same cell may also do the reverse process, which is endocytosis. And if that's the case, then the cell may not grow as much. Also, the vesicle is very tiny compared to the size of the cell. So, like that difference of expansion might not be very big. Anyways, Moving on, how do we actually put this in words, right? So this is the list of the cell structures involved. How do we talk about it? Number one, there's synthesis of protein in the ribosome or the RER. The transport vesicle buds off the endoplasmic reticulum and fuses with the Golgi body. There's modification of protein in the Golgi body and separation of secretory vesicle from the Golgi body can occur after that. And the vesicle can then travel to the cell surface membrane where it fuses, and this contents of the vesicle can then be secreted by exocytosis. Now, bear in mind that this process also works to embed a protein at the cell surface membrane. The only difference between a protein at the cell surface membrane and a secreted protein would be that in the vesicle, instead of the proteins being inside, this is a secreted protein, the protein would be on the surface of the vesicle. So when the secretory vesicle fuses with the cell surface membrane, the vesicle membrane also becomes part of the cell surface membrane, right? So that process, this cell surface membrane then becomes this cell surface membrane protein then becomes part of the cell surface membrane just by being at the phospholipid bilayer of the vesicle instead of the inside. Ta-da! That is the production and secretion of proteins. Let me give you a little exercise. This is a past year question. And it says, when a mucus is secreted from a globule cell in trachea, these events take place. This, and this. What is the sequence in which these events take place? Pause the video, think about it, and talk about it. Okay, by the way, mucus is made up of mucin, and mucin is a glycoprotein, which is a combination, a protein that is made out of carbohydrate, therefore glyco, and a protein. Haha. So that should give you a hint on how to answer this question. Pause the video. You done? Okay, so what is the sequence in which these events take place? The answer is 1, 4, 2, 3. So addition of carbohydrate protein, which happens at the Golgi or possibly the ER, doesn't matter. Number 2, after it's modified either in the Golgi or ER, there's separation of vesicle from the Golgi apparatus, right? This would be a secretory vesicle. Number 2, there's fusion of the secretory vesicle with the plasma membrane, and therefore secretion of the glycoprotein, which is mucin, out of the cell through a process called exocytosis. And that is the process of secreting protein from your cells. 3 more cell structures to go before this video ends. Don't forget that Golgi, in addition to making secretory vesicles, also is in charge of synthesizing lysosomes, which is actually a vesicle as well, but it's a special vesicle with hydrolytic enzymes or lysozymes. And the function of these enzymes are to break down unwanted structures via hydrolysis in an acidic environment. This vesicle has an acidic environment to kind of facilitate that process. Now, unwanted structures is allowed, worn out organelles is okay, or dead cells is fine, but the exam would reject, right? Means they will minus marks if you wrote dead organelles, because organelles are not living things. We don't see organelles as living, we see whole cells as living. So do not write dead organelles, write worn out or damaged organelles would be fine. So this is definitely not what they want. Right, worn out organelles, unwanted structures, or dead cells are accepted. Now, in white blood cells especially, they have plenty of lysosomes, and these lysosomes are able to digest bacteria. And you can see a picture of a white blood cell here. You can see how the bacteria is engulfed by this cell, and then the lysosome and the phagosome actually fuses, and this exposes the bacteria to the lysozyme, and the lysozyme breaks it down, and then like the cell debris actually gets thrown out the cell via exocytosis. Now, this process you will learn in more detail in the coming chapters. I know I say that a lot, but I really do explain them in more detail later on. Anyways, 2 more cell structures to go. Number 8, mitochondria. Now, we all know mitochondria is the powerhouse of the cell, but if you write it in exam in Cambridge, you'll get it wrong. Now, what should you write instead? Now, mitochondria synthesizes ATP in the form, energy in the form of ATP. Okay, it's not here yet. Let's look at some of the other details first. Now, it is a relatively large organelle. It's not as large as the nucleus. I think if you put them in order, nucleus is largest, followed by the chloroplasts, followed by the mitochondria. So, mitochondria. Mitochondria has double membranes as well. Actually, the nucleus, chloroplast, and mitochondria are friends. They all have double membranes. Mitochondria, though, in particular, has a special thing inside. It's called cristae, not to confuse with cisternae for Golgi and ER. This is cristae, which is folded inner membrane inside the mitochondria. And this increases surface area for a lot of protein and basically energy ATP synthesizing equipment. The interior solution is called a matrix, and it actually contains a lot of enzymes and also 70S ribosomes, which we discussed earlier, and also small circular DNA. You're like, huh? Small circular DNA? Okay, so it's still double helix DNA, but instead of like, a huge, like a linear strand, it kind of joins back on itself. So it's still a double helix, but this is a bad representation drawing, but it kind of loops back on itself. This would be circular DNA. This is linear. Now, this small circular DNA would code for some mitochondrial proteins that are needed for energy synthesis. Yeah. Okay. Mitochondria also divides by binary fission, which is quite cute. So it kind of cleaves down the middle and it separates it to become 2 here. And the reason for that weirdness in general is that mitochondria are hypothesized to have a prokaryotic origin. What does that mean? This means that maybe once upon a time, mitochondria, as well as chloroplasts that you will see later, actually were other living cells, maybe other living bacteria, which were able to synthesize their own energy in the form of ATP. Chloroplasts as well may have been a bacteria in the past that could harvest sunlight and convert it into energy and food. And basically, our ancestral cell, long time ago, were able to use these other bacteria in order to make energy in the form of ATP for itself, or harvest light to form food like plants do. So yeah, that's why it's kind of interesting that it has its own DNA, it has its own ribosomes, and it has double membranes. These are all some form of evidence that this bacterial hypothesis is true. Kind of interesting. So mitochondria, again, not powerhouse of the cells. Yes, it's powerhouse of the cell, but you can't write it that way. So how do you write it? The function is the site of aerobic respiration. It's to synthesize ATP or produce energy in the form of ATP. The reason why I keep saying ATP instead of just synthesizing energy is because they reject, as in their minus marks, if you write produce energy alone without mentioning ATP. Because energy can't be created or destroyed, right? Physics. So mitochondria don't produce energy. It just produces energy in a form of ATP, which would be able to be used easily by different reactions of our cell. Yeah. Now, aerobic respiration. What does this term mean? Now, when we say respiration, we often think of breathing in and breathing out. That's not respiration we're talking about. We are talking about the conversion of glucose, oxygen into carbon dioxide, water, and ATP. That is what we mean by aerobic respiration, breaking down food in order to form energy in the form of ATP. Again, please do not write these two sentences right here. Let's look at this electron micrograph. Again, this is because we need to do three things, right? And three things is name it, recognize it, and know its function. And also in a certain extent, to a certain extent, know its function and structure as well. So what are the things you see here that you could identify? What is A? What's B? What is C? Now A is the biggest organelle, which is the nucleus. This is a very zoom-in diagram. We know the nucleus because we see like these chromatin-like structures hanging about, and it's really big. It's not an other cell. This is inside a cell. Now this here is mitochondria. B is mitochondria. In the mitochondria, you can see here we have some inner membranes going on. These are cristae. And yeah, they are pretty large too in comparison. It's not as big as the nucleus, but it's still pretty large, okay? It's still pretty large. Now how about C? Now C is a little bit distant from the nucleus, right? So it's maybe not very continuous, but it does have kind of a rougher appearance. So I would like this. This is rough and this is kind of near. So this is probably the rough endoplasmic reticulum. And I would say that this would be the rough endoplasmic reticulum as well. It's just maybe continuous because it's a similar thickness, you see. But honestly, if you're not sure in an exam, you can easily write endoplasmic reticulum in general. If you're not sure if it's smooth or rough, and if it's really far away, then write Golgi. Usually, they will only ask you what is obvious, not what is debatable. Yeah, so that's the mitochondria. This is also the mitochondria. This is also the mitochondria. Get used to the diagrams, man. You can see the inner cristae looks beautiful. Sometimes it looks cylindrical, not cylindrical, like rod shape. That's what we call this shape, by the way, rod shape. And sometimes it looks circular, so oval, circular. What's up? Now, it's because you're probably seeing it differently. One is a longitudinal cross-section, one is like a transverse section. Also, shape of mitochondria may vary, and maybe like you're viewing it from different angles, or maybe it's just divided. Right, it's just divided by binary fission, so you can't really tell the shape properly. Like the shape is not fixed. So yeah, it's mitochondria, and this is actually a common question in past years. Moving on to our last organelle for this video. Chloroplasts. We saw some pictures of chloroplasts before, but here is a little representative diagram. It is also a relatively large organelle, second in place after the nucleus. It's definitely visible under the light microscope. It is oval shaped. It is two membranes. It has chlorophyll. It has thylakoid. These thylakoids are flattened membrane stacks, and multiple thylakoids stacked together is called grana. And this grana is in a liquid of called in a liquid inside called stroma, not inferior solution, interior solution. I was reading it wrong in my brain. Anyways, grana, stroma, right? Grana again is thylakoid stacks, stroma is the fluid. Now, just like mitochondria, it's special, right? Again, it's two membranes. It contains only as ribosomes, small circular DNA, and because chloroplasts harvest light energy to make food, they also have starch grains inside chloroplast. It also divides by binary fission, and all of this is because, well, it probably has a prokaryotic origin. This is chloroplasts in real electron micrograph diagrams. You can see the grana here, the thylakoid stacks. It looks beautiful, don't you think? Now, under light microscope, we can definitely see this as well, and they will appear green because of the chlorophyll. Now, here is a plant cell, and you can see the kind of contrast in size between everything else. We can see the nucleus, which is the biggest organelle usually. Now, the vacuole is full of water at this point, so it looks a little bit bigger than nucleus. Quite common in plant cells, but not all. But vacuoles are not usually visible under the light microscope. So nucleus is still the most visible, largest organelle of plants. Anyways, you can see that next up, like the next biggest thing would be the chloroplasts, and they are huge, don't you think, in comparison to everything else? And we can see the mitochondria here being slightly smaller than the chloroplasts, and the endoplasmic reticulum isn't even very seen. The Golgi apparatus is barely visible there. And that's because this image is probably not high resolution enough and doesn't have enough of a magnification to visualize those fine membranes. But yeah, look how big chloroplasts are, right? Okay, let's talk about the function a little bit. Now, the function, obviously, is the site of photosynthesis. Now, this may come as a surprise because there are two main processes in photosynthesis: the light-dependent reaction, and this is where light energy is absorbed and water is used to synthesize ATP. So surprise, surprise! Mitochondria is not the only powerhouse of the cell, especially in plants, right? Because the chloroplast is able to synthesize ATP as well. But this ATP is used inside the chloroplast to convert carbon dioxide into glucose in a light-independent reaction. The conversion of carbon dioxide into glucose is called carbon fixation. And you don't need to think about this right now, this terms like dependent and like independent. You just need to know that it synthesizes ATP. So photosynthesis, and what it does is to convert carbon dioxide into glucose. So only the red and bold words here. However, you will learn these processes in more detail in A2, chapter 13, to be specific. Wait, yes, chapter 13. So yes, that is the chloroplast, and I think we are done with 9 different structure. Whew, a long video, lots of content. I hope that was helpful though. Um, all of the essential information are in red and bold, just in case you don't know. And these are all based on mark schemes. If you'd like a copy of my notes, they will be available online. Look at the video description for more information. See you next time. Bye-bye.